Greetings everyone, this is Raul speaking, and welcome to the second video in our professional development installment. This week's professional development video is dedicated to critically evaluating scientific figures. I would like to begin this video by prefacing it with two key points. First, your ability to critically evaluate a figure and the time that it takes you to do so improves with experience, meaning that this is a skill that requires practice. Second, your ability to critically evaluate a figure improves as you develop a stronger background regarding a particular scientific topic or technique, meaning that this is a skill that requires context. In this video, I hope to quickly equip you with some of the fundamental techniques necessary to kickstart your scientific journey. Before you begin to evaluate a specific figure, there are two things that you need to keep in mind, and these will help you critically approach the task. First, it is important to keep in mind the greater scientific question of the specific research paper that you are reading. This will help you put all the findings into context with each other and give you insight into the steps the authors took experimentally to test their hypotheses. The second thing to keep in mind is what is the lesser point of the specific figure that you are about to dig through. In other words, what is it trying to convince you of? And as a hint, it is often the figure heading itself. For this figure that we will use as an example, the authors want to convince us that norepinephrine excites action potentials in Merkel cell afferents. For some context here, afferents is referring to the axons from the dorsal root ganglia that synapse with Merkel cells in the epidermis. As you start to review a figure, here are some key points to keep in mind. First, does the panel that you are looking at consist of raw data? Raw data being something that can be quantified, but in that specific panel or instance, it hasn't been. This can be an image or an electrophysiology trace. Next, you can ask yourself, do the scale bars seem legit? After some time familiarizing yourself with a specific type of data, such as electrophysiology recordings, comparing the raw data to the scale bars can give you a sense for the quality of the recordings. Next, you can ask yourself, is the raw data representative or just an example? The point here being that we need to decide visually if the raw data is in line with the summary data. If it is a representative image or trace, it should reflect the summary data. If it doesn't, it is likely just an example. And an ugly truth is that papers often only show one example, and it is human to want to pick the best example. If the panel you are looking at is not raw data, then it is likely summary data, data that has been analyzed and graphed in an attempt to clearly communicate a result. Here, you'll want to know what type of graph are you looking at. You will learn over time that there are specific things to keep in mind when you are looking at a bar graph versus a scatter plot or a histogram or a line plot. You'll want to know what is being plotted across the different axes and that these axes make sense, meaning that they are not arbitrarily cut off in any way that could bloat differences between variables. At this point, you'll also want to make observations about the data and draw your own conclusions. Some final remarks that I want to make regarding key points while critically evaluating a figure are that nowadays Most figures often have a panel dedicated to summarizing the experimental approach, and if you get stuck, you can always dive into the figure legend, the results section, and the methods section for clarification. This content is always important, but you will find it to be extra helpful in the beginning. Now, let's take some of the key points that we have discussed here and go through an example figure. To give you some context, the figure that we are about to evaluate comes from data acquired using the skin nerve prep, depicted here on the upper right hand corner. In this preparation, electrophysiological recordings are made from axons that innervate the epidermis. In this experiment, the authors are recording from axons that form synapses with Merkel cells. The point that they are trying to make is that norepinephrine excites action potentials in Merkel cell afferents. So let's see if we come to the same conclusion. In panel A, we are being presented with raw data, specifically an electrophysiology trace. I know from experience, but also from the figure legend, that these are traces of action potential firing. It seems like they recorded from these axons after they were treated with three different neurotransmitters: norepinephrine, serotonin, and dopamine. From this raw data, we can already appreciate that treatment with norepinephrine causes a dramatic increase in action potential firing. Additionally, on the right half of panel A, the waveform of action potentials in the norepinephrine group matches that of those evoked by touch. This is not true in the other two experimental conditions. Let's see how this data looks like when we look at the distribution of results in panel B. Panel B is a scatter plot demonstrating the response to neurotransmitter treatment across several recordings in several animals. This information is in the figure legend, but let's also break it down ourselves. On the x-axis, we have the results from treatment with three different neurotransmitters at three different concentrations. On the y-axis, we have the amount of action potentials fired in a 10-minute trace. Judging from the 2-minute scale bar in panel A, I would assume that the total length of the sample traces is 10 minutes. So in panel B, they are quantifying many different instances of the data depicted in panel A. It is immediately apparent, as it is with the raw data, that treatment with 5 millimolar of norepinephrine evokes a dramatic increase in action potential firing. The example trace in panel A is that of treatment with 5 millimolar, according to the figure legend. So it seems like in this instance, the trace in panel A is very representative of the finding in figure B. What is also interesting is that the lower concentrations of norepinephrine don't evoke the same response, meaning that there seems to be a threshold for activation. The last point is better appreciated in panel C, where the authors plot a dose-response curve. The response is the y-axis, and it is the amount of action potentials fired in a 10-minute trace, plotted against the dose, treatment with norepinephrine at various concentrations. This figure characterizes the relationship between neurotransmitter treatment and action potential firing. So our conclusion from this figure is that treatment with norepinephrine as compared to serotonin and dopamine elicits robust action potential firing. After you have reviewed the figure, you can compare and contrast your conclusions with those of the authors. You can also ask yourself, what are some of the missing pieces of information? And to flex and test your knowledge, you can come up with experiments that could fill those gaps. In the interest of time and keeping this video short, we did not cover data distributions or statistical testing. but we plan on doing so in future videos. For now, remember that the internet and your colleagues are a great resource for when you get stuck. I want to end this video with a cautionary anecdote. Always be skeptical of any summary figure or graph that does not show you what the underlying data is. Bar charts or box plots with no data points can be notoriously deceptive. Thanks for listening.